Hello everyone, we are the facilitator for the chapter 3 and we're going to learn about the anatomy of the nervous system. For our chapter outline, we are going to tackle upon general layout of nervous system, cells of the nervous system, neuroanatomical techniques and direction, and anatomy of the central nervous system. So the divisions of the nervous system. It consists of two parts, the central nervous system, which is for short called CNS, and peripheral nervous system, which is for short called PNS. So the central nervous system consists of brain and the spinal cords, while peripheral nervous system consists of parts outside the brain and the spinal cord, connects the CNS to the limbs and the organs to help regulate. So for this part, we're going to focus on the PNS part. So PNS has two parts of system, which is the somatic and the autonomic nervous system. Somatic nervous system is the part of the peripheral nervous system associated with the voluntary control of body movements via skeletal muscles. The somatic nervous system consists of afferent nerves or sensory nerves and efferent nerves or motor nerves. The autonomic nervous system is a control system that acts largely unconsciously and regulates bodily functions, such as the heart rate, digestion, respiratory rate, pulmonary response, urination, and sexual arousal. Afferent nerve signals from the internal organs to CNS. Efferent nerves carry motor signals from the CNS to the internal organs. Two kinds of nerves, the sympathetic nerves and parasympathetic nerves. Sympathetic nerves project from the CNS and the lumbar and thoracic regions of the spinal cord, while parasympathetic nerves project the brain and the sacral region of the spinal cord. Sympathetic nerves prepares the body for the fight or flight response during any potential danger, while parasympathetic nerves inhibits the body from overworking and restores the body to a calm and composed state. The cranial nerves are pairs of nerves that connect your brain to the different parts of your head, neck, and trunk. There are 12 of them, each named of them for their function or structure. Each nerve also has a corresponding Roman numeral between 1 to 12. The meninges refer to the membranous covering of the brain and the spinal cord. There are three layers of meninges known as the dura mater, arachnoid mater, and pia mater. These coverings have two major functions, provide a supportive framework for the cerebral and cranial vasculature. Ventricles and cerebrospinal fluid. Cerebrospinal fluid or CFS protects the central nervous system, fills the subarachnoid space, the central canal of the spinal cord, and the cerebral ventricles of the brain. It supports and cushions the brain. The central canal, also known as epidermal canal, is the cerebrospinal fluid filled space that runs through the spinal cord. The central canal below at the ventral system of the brain from which it receives cerebrospinal fluid and shares the same epidermal lining. Cerebral ventricles are four interconnected cavities of the brain that lined by epidermal cells and filled by cerebrospinal fluid. Choroid plexus, a network of blood vessels in each ventricle of the brain, derived from the pia mater and produces the cerebrospinal fluid. Dural sinuses are venous channels found between the endosteal and meningeal layers of dura mater in the brain. The blood-brain barrier helps block harmful substances such as toxins and bacteria from entering the brain, but scientists knew that the brain also depends upon the delivery of hormones and key nutrients, including glucose and several amino acids from other organs of the body. And now we're going to talk about the cells of the nervous system. Neurons. Specialized cells for the reception, conduction, and transmission of electrical signals. Neuron cell membrane composed of lipid bilayers, which has two layers of fat molecules, which is the channel proteins and the signal proteins. A channel protein is a specialized arrangement of amino acids which embeds in the cell membrane, providing a hydrophilic passageway for the water and small polar ions. Signal protein transfer a signal to the inside of a neuron when particular molecules bind them on the outside of the membrane. External feature of a neuron. Cell membrane encloses the neuron. Dendrites receive most of the synaptic contacts from other neurons. Axon hillock, cone-shaped, junction between the axon and the cell body. Axon, long narrow process that projects from the cell body. Cell body, metabolic center of a neuron also called soma. Myelin, fatty insulating around many axon. Nodes of Ranvier, gaps between sections of myelin. Botons, button-like ending of axon branches. Release chemicals into synapses. Synapses, gaps between adjacent neuron of which chemical signals are transmitted. Internal features of a neuron. Mitochondria, sites of aerobic, which means oxygen consuming, energy release. Nucleus, a spherical DNA containing structure of a cell body. Endoplasmic reticulum, a system of folded membranes in the body. Rough portions, those with ribosomes, plays a role in synthesis of proteins. Smooth portions, those without ribosomes, plays a role in the synthesis of fats. Cytoplasm, the clear internal fluid of the cells. Ribosomes, internal cellular structure on which proteins are synthesized. They are located in the endoplasmic reticulum. Golgi complex, a connected system of membranes that packages molecules in the vesicle. Microtubules, tubules responsible for the rapid transport of material throughout neurons. Synaptic vesicles, a spherical membrane packages that stores neurotransmitter molecules ready for release near synapses. Neurotransmitters, molecules that are released from active neurons and influence the activity of other cells. Classes of neuron. Multipolar neuron. A multipolar neuron is a type of neuron that possesses a single axon and many dendrites, allowing for the integration of a great deal of information from other neurons. These processes are projections from the neuron cell body. A unipolar neuron in which one process called a neurite extends from the cell body. The neurite then branches to form dendritic and axonal processes. Most neuron in the central nervous system of invertebrates, including insects, are unipolar. Bipolar neuron is a type of neuron that has two extensions. Many bipolar cells are specialized sensory neurons for the transmissions of sense, such as they are part of sensory pathways for smell, sight, taste, hearing, touch, and balance. Interneurons are the central nodes of neural circuits enabling communication between sensory or motor neurons and the central nervous system. They play vital roles in reflexes, neural scoliosis, and neurology in the adult mammalian brain. Now we have reached the part that we're going to tell you the story of glia, the forgotten cell. So there are different kinds of glia. First is the oligodendrocytes, which is the extensions that wrap around the axons of some neurons of the CNS. Under oligodendrocytes is myelin, a fatty insulating substance. Myelin sheaths forms increase the speed and efficiency of the axonal conduction. Schwann cell, second class of glia. It guides axonal regeneration after damage. Microglia, third class of glia, responds to injury or disease by multiplying, engulfing cellular debris or even entire cells, trigger inflamed responses. Astrocytes, fourth class of glia, which is the largest, allow passage of some chemicals from the blood into the CNS neurons and in blooding other chemicals. Ability to relax blood vessels. Neuroanatomical techniques and directions. So this is the part of the lessons which we are going to tackle upon how cells became visible into our eyes. So first is the Golgi stain, which is invented by Camillo Golgi, which exposed a block of neural tissues to potassium dichromate and silver nitrate. The only problem with Golgi staining is that it only stains, it doesn't really show us proper details of the neuron. So second is the Nisil stain, which is invented by Franz Nisil. It uses crystal violet. First neural staining procedure. Penetrates all cells on the slide. As you can see in the picture, the cells became violet because of the crystal, and it shows some details that Golgi stain cannot. Lastly is the electron microscopy. Provides info about the details of the neural structure. Electron micrograph equals result. Scanning electron microscope equals 3D result, which is visible to the given picture. Neuroanatomical tracking techniques. First is the anterograde tracing methods, used when an investigator wants to trace the paths of axons projecting away from cell bodies located in a particular area. While retrograding tracing methods works in reverse. They are used when an investigator wants to trace the paths of axon projecting into a particular area. Directions in the vertebrate nervous system. You need to refer to the picture and listen carefully. Anterior posterior is 1, dorsal ventral is also 1, medial and lateral is also 1. So anterior is towards the nose end, which is also called rostral. Posterior towards the tail end, which is also called caudal. Dorsal ventral. Dorsal towards the surface of the back or the top of the head, while ventral towards the surface of the chest or the bottom of the head. Medial lateral. Medial towards the midline of the body, lateral away from the midline of the body. Please refer to the picture attached.
The anatomy of the central nervous system. The spinal cord is comprised of two different areas: gray matter, composed largely of cell bodies and unmyelinated interneurons, and the white matter, composed largely of myelinated axons. The myelin, which is a lipid-rich substance, gives the white matter its glossy white sheen. The two dorsal arms of the spinal gray matter are called the dorsal horns, and the two ventral arms are called the ventral horns. The dorsal horn is comprised of sensory nuclei that receive and process incoming somatosensory information, while the ventral horns contain the cell bodies of motor neurons that send axons via the ventral roots of the spinal nerves. Pairs of spinal nerves are attached to the spinal cord, one on the left and one on the right, at 31 different levels of the spine. Each of the 62 spinal nerves divides as it nears the cord, and its axons are joined to the cord via one of the two roots, the dorsal root and the ventral root. The dorsal root, whether somatic or autonomic, are sensory unipolar neurons, with their cell bodies grouped together just outside the cord to form the dorsal root ganglia. Many of their synaptic terminals are in the dorsal horns of the spinal gray matter. On the other hand, the neurons of the ventral root are motor multipolar neurons with their cell bodies in the ventral horns. Those that are part of the somatic nervous system project to skeletal muscles, and those that are part of the autonomic nervous system project to the ganglia, where they synapse on neurons that in turn project to internal organs, such as heart, stomach, and the liver. The five major divisions of the brain. To understand why the brain is considered to be composed of five divisions, it is necessary to understand its early development. In the vertebrate embryo, the tissue that eventually develops into the central nervous system is recognizable as a fluid-filled tube. The first indications of the developing brain are three swellings that occur at the anterior end of this tube: the adult forebrain, midbrain, and the hindbrain. Before birth, the initial three swellings in the neural tube become five. This occurs because the forebrain swelling grows into two different swellings, and so does the hindbrain swelling. From anterior to posterior, the five swellings that compose the developing brain at birth are the telencephalon, the diencephalon, the mesencephalon, the metencephalon, and the myelencephalon. Myelencephalon, also known as the medulla, is the most posterior division of the brain. It is composed largely of tracts carrying signals between the rest of the brain and the body. One interesting thing about the myelencephalon is the reticular formation, which is a complex network of about 100 tiny nuclei that occupies the central core of the brainstem from the posterior boundary of medulla to the anterior boundary of the midbrain. It is sometimes called the reticular activating system, because parts of it seem to play a role in arousal. However, the various nuclei of the reticular formation are involved in a variety of functions, such as sleep, attention, movement, the maintenance of muscle tone, various cardiac, circulatory, and respiratory reflexes. The metencephalon, which houses many ascending and descending tracts and part of the reticular formation, consists of two main parts. The bulge on the brainstem's ventral surface is called the pons, which contains tracts that carry signals from the cerebrum to the medulla and to the cerebellum. The cerebellum coordinates motor activity and helps regulate muscle tone. It is a large convoluted structure on the brainstem's dorsal surface and an important sensory motor structure. Cerebral damage eliminates the ability to precisely control one's movements and to adapt them to changing conditions. The fact that the cerebral damage also produces a variety of cognitive deficits, such as deficits in decision-making and in the use of language, suggests that the function of the cerebellum are not restricted to sensory motor control. The mesencephalon, also known as the midbrain, has two divisions. One is the tectum, which is the dorsal surface of the midbrain. In mammals, it is composed of two pairs of pompes. The inferior colliculi have an auditory function, and the superior colliculi have a visual motor function, specifically to direct the body's orientation toward or away from particular visual stimuli. In lower vertebrates, it is entirely visual motor and sometimes referred to as the optic tectum. Another one is the tegmentum, which is the division of the mesencephalon ventral to the tectum. In addition to reticular formation and tracts of passage, tegmentum contains three colorful structures: the periaqueductal gray, which is the gray matter situated around the cerebral aqueduct, which is the duct connecting the third and fourth ventricles. It plays a critical role in autonomic function because it motivates behavior and behavioral responses to threatening stimuli. The substantia nigra and red nucleus are both important components of the sensory motor system. The diencephalon, which acts as a primary relay and processing center for sensory information and autonomic control, is composed of two structures: the thalamus and the hypothalamus. The thalamus is the large two-lobe structure that constitutes the top of the brainstem. One lobe sits on each side of the third ventricle, and the two lobes are joined by the massa intermedia, which runs through the ventricle. Visible on the surface of the thalamus are white lamina layers that are composed of myelinated axons. The thalamus comprises many different pairs of nuclei, most of which project to the cortex. The most well understood thalamic nuclei are the sensory relay nuclei, which is the nuclei that receive signals from sensory receptors, process them, and then transmit them to the appropriate areas of sensory cortex. The hypothalamus is located just below the inferior thalamus. It plays an important role in the regulation of several motivated behaviors, such as eating, sleep, and sexual behavior. It exerts its effects in part by regulating the release of hormones from the pituitary gland, which dangles from it on the ventral surface of the brain. The literal meaning of pituitary gland is nut gland. In addition to the pituitary gland, Two other structures appear on the inferior surface of the hypothalamus. The optic chiasm is the point at which the optic nerves from each eye come together. The X shape is created because some of the axons of the optic nerve decussate, which means cross over to the other side of the brain via the optic chiasm. The decussating fibers are said to be contralateral, or projecting from one side of the body to the other, and the non-decussating fibers are said to be ipsilateral, which means staying on the same side of the body. The mammillary bodies, which are often considered to be part of the hypothalamus, are a pair of spherical nuclei located on the inferior surface of the hypothalamus, just behind the pituitary gland. The telencephalon, also known as the cerebrum, is the largest division of the human brain, which mediates the brain's most complex functions. It initiates voluntary movement, interprets sensory input, and mediates complex cognitive processes such as learning, speaking, and problem-solving. The cerebral hemispheres are covered by a layer of tissue called cerebral cortex, which is mainly composed of small unmyelinated neurons and is often referred to as the gray matter. In contrast, the layer beneath the cortex is mainly composed of large myelinated axons, often referred to as the white matter. In humans, the cerebral cortex is deeply convoluted or furrowed, which increases the amount of cerebral cortex without increasing the overall volume of the brain. Not all mammals have convoluted cortexes. Most mammals are lissencephalic or smooth-brained. The large furrows in a convoluted cortex are called fissures, and the small ones are called sulci. The ridges between fissures and sulci are called the gyri. The cerebral hemisphere are completely separated by the largest of the fissures, called the longitudinal fissures. The cerebral hemisphere are directly connected by a few tracts spanning the longitudinal fissures. These hemisphere connecting tracts are called cerebral commissures. The largest cerebral commissure is the corpus callosum, which is the largest cerebral commissure. It connects the two halves of the brain and delivers messages from the one half of the brain to the other. There are two major landmarks on the lateral surface of each hemisphere, the central fissure and the lateral fissure. Both of these features partially divide each hemisphere into four lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. Among the largest gyri are placental gyri, found in frontal lobe, postcentral gyri, found in parietal lobe, and the superior temporal gyri, found in temporal lobes. Take note that the cerebral lobes are not functional units. It is best to think of the cerebral cortex as a flat sheet of cells that just happen to be divided into lobes because pressures cause it to be folded in on itself at certain places during development. The occipital lobe is the brain's visual cortex, which allows a human not only to see and process stimuli from the external world, but also to assign meaning to and remember visual perceptions. We humans rely heavily on the analysis of visual input to guide our behavior, and the occipital cortex and large areas of adjacent cortex perform this function. The parietal lobe has two large functional areas: the postcentral gyrus, which analyzes sensation from the body, and the remaining areas of cortex in the posterior part of the parietal lobes play roles in perceiving the location of both objects and our own bodies and in directing our attention. The cortex of each temporal lobe has three general functional areas. The superior temporal gyrus is involved in hearing and language. The inferior temporal cortex identifies complex visual patterns, and the medial portion of temporal cortex, which is not visible from the usual side view, is important for certain kinds of memory. And lastly, each frontal lobe has two distinct functional areas. The precentral gyrus and adjacent frontal cortex have a motor function, whereas the frontal cortex anterior to motor cortex performs complex cognitive functions such as planning response sequences, evaluating the outcomes of potential patterns of behavior, and assessing the significance of the behavior of others. A About 90% of human cerebral cortex is called neocortex. It is a six-layered cortex of relatively recent evolution. The layers of neocortex are numbered numeral 1 to numeral 6 starting at the surface. In the diagram, it is apparent that three important characteristics are present in the neocortex. The first important characteristic is that many cortical neurons fall into one of two different categories. The pyramidal cells are large multipolar neurons with pyramid-shaped cell bodies, a large dendrite called apical dendrite, and a very long axon. The second one is the stellate cells, which are small star-shaped interneurons or neurons with a short axon or no axon. The second important characteristic is that the six layers of neocortex differ from one another in terms of the size and density of the cell bodies and the relative proportion of pyramidal and stellate cell bodies that they contain. The third important characteristic states that many long axons and dendrites course vertically through the neocortex. This vertical flow of information is the basis of the neocortex's columnar organization. The fourth important characteristic of neocortical anatomy is not apparent in the diagram. Although neocortex is six-layered, there are variations in the thickness of the respective layers from area to area. For example, because the stellate cells of layer 4 are specialized for receiving sensory signals from the thalamus, layer 4 is extremely thick in areas of sensory cortex. Conversely, because the pyramidal cells of layer 5 conduct signals from the neocortex to the brainstem and spinal cord, layer 5 is extremely thick in areas of motor cortex. The hippocampus is one important area of cortex that is not neocortex. It has only three major layers: CA1, CA2, and CA3. These three major layers form the trilaminar loop, which is the processing center of long-term memory. It is located at the medial edge of the cerebral cortex as it folds back on itself in the medial temporal lobe. Hippocampus plays a major role in some kind of memory, particularly memory for spatial location. Although much of the subcortical portion of the telencephalon is taken up by axons projecting to and from the neocortex, there are several large subcortical nuclear groups. Some of them are considered part of either the limbic system or the basal ganglia system. The limbic system is a circuit of midline structures that circle the thalamus. It is involved in the regulation of motivated behaviors, including the four F's of motivation: the fleeing, feeding, fighting, and sexual behaviors. Major structure include the amygdala, the fornix, and the cingulate cortex, and the septum. The limbic circuit starts at the amygdala, which is the almond-shaped nucleus in the anterior temporal lobe. Posterior to the amygdala is the hippocampus, which runs beneath the thalamus in the medial temporal lobe. Next is the cingulate cortex, which is a large strip of cortex in the cingulate gyrus on the medial surface of the cerebral hemispheres that encircles the dorsal thalamus. Next, fornix also encircles the dorsal thalamus. It leaves the dorsal end of the hippocampus and sweeps forward in an arc coursing along the superior surface of the third ventricle and terminating in the septum and the mammillary bodies. Lastly is the septum. It is a medullary nucleus located at the anterior tip of the cingulate cortex. Several tracts connect the septum and the mammillary bodies with the amygdala and hippocampus, thereby completing the limbic ring. The basal ganglia play a role in the performance of voluntary motor responses and decision-making. It also starts in the amygdala, which is considered part of both systems. Sweeping out of each amygdala first is a posterior direction, and then in an anterior direction is the long tail-like caudate. Each caudate forms an almost complete circle. In its center, connected to it by a series of fiber bridges, is the putamen. Together, the caudate and the putamen, which both have a stripe appearance, are known as the striatum or the stripe structure. The remaining structure of the basal ganglia is the pale circular structure known as the globus pallidus. The globus pallidus is located medial to the putamen between the putamen and the thalamus. Another part of the basal ganglia that is of particular interest to biopsychologists is the nucleus accumbens, which is the medial portion of the ventral stratum. The nucleus accumbens is thought to play a role in the rewarding effects of addictive drugs and other reinforcers. The basal ganglia play a role in the performance of voluntary motor responses and decision-making. Of particular interest is the pathway that projects to the stratum from the substantia nigra of the midbrain. Parkinson's disease, a disorder characterized by rigidity, tremors, and poverty of voluntary movement, is associated with deterioration of this path. This table is a summary of the major brain structures. To assess how much you've learned from this chapter, try to label the parts of the brain. The answers will be shown in the next slide.